Today is April 10th, 2018. My name is Martin Frank, and I'm at the Medical College of Wisconsin interviewing Dr. Alan W. Cowley Jr. for the Society's Living History program. Dr. Cowley has been an APS member since 1972 and has been affiliated with the Medical College of Wisconsin since 1980. Dr. Cowley's research has focused on the role of baroreceptor reflexes, the renin-angiotensin system, and vasopressin in both the short and long-term regulation of arterial blood pressure. During the past decade, he has pioneered efforts to attach systems-level biology to the genome, providing a novel insight into the location of genes that underlie complex disease, planting the seeds for this field now referred to as physiological genomics. Alan, welcome to the Living History Project, and I thank you for agreeing to be interviewed for the series. If you are ready, I would like to ask you a few questions about your career.
Thank you, Marty. It's an honor to be here and to be interviewed by you.
Well, I'm glad to hear it. Let's start by asking about your family upbringing and how you became interested in science.
Well, that's a long story, but
well, we have plenty of time.
I will start in my youth. I was very fortunate to have been adopted as an infant just at the beginning of World War II by a wonderful family. My father was a physician, general family physician at the time, who was one of the early pioneers in applied cardiology. My mother was trained as a nurse, and they had met at the University of Pennsylvania when he was a resident, and moved to Harrisburg, where I grew up. Harrisburg, Pennsylvania, till I was 10, and then we moved out of the city and into a suburb across the Susquehanna River, Camp Hill. So I was raised in a physician's family. I would spend holidays in particular. This is the time when physicians made house calls. And I would go along with Dad when he was called in the middle of Christmas dinner on an emergency. And he'd take me along. Sometimes there were sad events. Other times just indigestion. But that was sort of the way I grew up. This was medicine and science, I must say, were part of my upbringing. Dad always had a lot of scientific books around. One of them, interestingly enough, happened to be Arthur Guyton's textbook of physiology and his monographs. As I went along and was in college, I found these sitting on his shelf. He would take me to the farm, local farm. He was a farm boy. He was a rancher from southern Utah, one of a large Mormon family. He sort of left the fold. His mother died when he was early in college, and she wanted him to be the one in the family that would be educated. With her untimely death, he made a vow to do that. Went to college, got into medical school at the University of Utah. It was a 2-year school at that time. They had agreement with the University of Pennsylvania, so he went east to finish medical school, met my mother, a nursing student there, and remained in Pennsylvania where I grew up. So it was a family rich with, my mother came from a teaching family. My grandfather was a high school principal, and my father was sort of the first in the family to really formally get an education from his side. So I must say they imprinted very strong principles upon me. And it wasn't necessarily being pushed in any direction, although I always felt when I was young that he expected to be, was hoping I'd follow in his footsteps as a physician. I chose not to do that for probably the very principal reason because he was considered just a paradigm of a physician in central Pennsylvania, and I thought I could never follow in his footsteps, and I sort of rebelled in school and in college and decided I ended up with a degree in economics, and that was a bit peculiar. And it turns out to be a mistake, because once I graduated from Trinity College, this was in Hartford, Connecticut, I got the usual family question, now what are you going to do with your economics degree? Well, I didn't want to go to graduate school. I didn't think I wanted to be a teacher of economics. So Dad says, well, you know, they're opening up the first branch bank in Harrisburg. He says, a patient of mine happens to run the bank, George Riley. He says, go talk to George and see if he can use somebody with an economics major. And I did, and I went to work in the bank. At the end of the week, Dad asked how it was going, and I said, this isn't going to work. I am never going to be a banker. And so he said, well, you have to do something. This is how I got into science. So,
how'd you decide to go and get a master's and a PhD in physiology?
well, this is
Obviously,
this
this
is the
contributed.
story.
Yeah.
This was the parental influence. So Father says, look, he said, you can live at home one more year, but you're going to go to college and take some additional courses while you're thinking about what you're going to do if it's not something in economics. And Dickinson College was about 20 miles up the road in Carlisle. So I signed up for what I thought would just be filler courses. One was an elective in biology. And you were supposed to have a project. And I came home and Dad says, well, what's your project going to be? And I said, I have no idea. And he said, you know, there's a drug that Merck has just made called MER-29. And it's just gone through its animal testing and some clinical trials, and they think it's causing cataracts and people's hair's falling out, and there's some issues with it. He said, why don't we get some rats and feed them some high-fat diet and put one group on MER-29 and have a group of controls and this and that. And I said, okay, let's get some rats.
And you did this in your house?
No, actually there was a little, truly a garage out behind the biology department.
Behind the biology department.
Yeah, behind the biology department. And some cages.
Okay.
And the biology professor said, fine, that's an interesting project. And I can't remember who bought the rats. Probably we did. But the rats did the project. Well, what was remarkable, and this was characteristic of my dad, he was very subtle. He just exposed you to things and then you could make up your mind. Well, I very soon found myself trying to figure out what this drug was doing. And I became very curious. I had taken my biology courses in chemistry. I actually had a minor in chemistry in college. I didn't enjoy biology in college. It was poorly taught and very boring, I thought. Just a lot of memorization. But I had enough to read the books. I knew where to look for things, and suddenly I found myself studying the cholesterol biochemical pathway, synthesis and degradation of cholesterol, because I wanted to see where this MER-29 was acting. That was really what got me going. Suddenly I found myself, for the first time in my life, independently trying to answer some questions. And I'd be darned if these rats didn't all go blind. Hair fell out.
All the ones on MER?
All the one on the MER-29. Our friend and neighbor was a pathologist. He said, well, let's look at the livers and do a little pathology on these rats. So he did the sections and...
it was your first research collaboration, right?
It was my first research collaboration. And he taught me the histology related to liver disease. They all got big fatty livers, a buildup of desmosterol, which because it blocked the conversion of desmosterol to cholesterol. So cholesterol levels fell and desmosterol levels went sky high and everybody got sick. So anyway, long story short, that's what got me interested. So at the end of the semester, Dad says, well, if that's the most I've seen you this excited about anything outside of playing tennis, and which I did enjoy doing. So he called a friend in Philadelphia who was the chairman of medicine, Dr. John Moyer at Hahnemann Medical School, and asked if there was a job for a kid wandering around trying to figure out what to do with his life. And Dr. Moyer stuck me in the Department of Medicine nephrology lab. And then I really got interested. That was the start of my career, and I worked with, that is a story in itself, because he put me in with a Japanese fellow who was spending 2 years on a research fellowship there, who spoke no English, and of course I spoke no Japanese, and we were just living together in this lab doing experiments together, lonely in Philadelphia. I didn't know anybody in Philadelphia, and so we befriended each other. He taught me an enormous amount of renal physiology during that year, drawing and conversing as best we could. Became a lifelong friend, went back to Japan, became the head of nephrology at Keio University in Tokyo. And I've had that followed up with probably 8 Japanese fellows that have gone through my lab from Keio and from Sendai, which I also developed a close relationship with. So I've had this long, really wonderful relationship with Japanese physicians and scientists over the years from that incident. And at the end of the year, I asked the chairman of physiology, Dr. John Scott, if I could, if he'd take me as a graduate student. He was skeptical. He said, well, let's try a master's first, see how you do. I did fine, enjoyed it, went on to get my PhD with Dr. Scott, who was a wonderful mentor, strict disciplinarian, rigid, very serious New Englander, which is just what I needed at that phase of my life. And that's how I got into all this.
What was your project with Dr. Scott?
He was interested at that time in the coronary circulation, and he had been doing a lot of anesthetized dog work, catheterization, placing catheters, doing washout techniques to look at coronary flow. And about the same time, the electromagnetic flow meter had been developed, and that happened to be going on. One of the 2 places it was going on. One was at Walter Reed, Donald Gregg, and a guy named Curry, Don Curry.
Fitzroy Curry?
Curry, I forget Curry's first name, but anyway, Curry was the engineer.
Okay. And
Gregg was, at that time, the prima donna of coronary circulation and physiology. And so I drove down to Bethesda with Dr. Scott, and Curry had made a flow meter and had made some handmade flow probes. And we had a wonderful surgical technician that worked in the lab right next to us, a fellow named Gomez, who had been the surgical technician of Charles Bailey, the fellow that developed the blue baby operation, who worked at Hahnemann. Bailey had just retired, and Gomez was looking for something to do. So Scott brought him into the lab to show me how to do surgical recovery, preparation and recovery. And so I worked with Gomez for half a year, learned how to instrument these dogs. And so this gets into another issue. I was really looking at the effects of catecholamines on intermediate metabolism in the heart. And that's what the project was about. So I instrumented with coronary flow probes, aortic flow probe, looked at cardiac output, left ventricular blood flow, coronary sinus vein instrumentation, and looked at the effects of catecholamines on free fatty acid metabolism and oxygen consumption, and infused labeled free fatty acids. The guy I did that with was John Spitzer, who was a young professor in the department at the time. He and Judy Spitzer were part of the department, and John taught me all the intermediate metabolism and the techniques for measuring free fatty acids, and so I really was co-mentored by John Scott and John Spitzer, and remained a lifelong friend of John and Judy Spitzer as he became the chairman of physiology at LSU.
Alan, you mentioned earlier that one of the books on your father's shelf was Guyton's book. And for those of you who don't know, you went down into Jackson, Mississippi to become an instructor in the Department of Physiology at University of Mississippi, working with Arthur Guyton.
I did.
And clearly, tell us about how you ended up there. And obviously, Arthur Guyton is one of the great names in our discipline, and what he was like, because he was a unique individual, and many people never had the experience of meeting him.
Well, this was another one of these fortunate turns in my life. I've had a lot of good fortune and been surrounded by some wonderful mentors. The way I got to Jackson was not a straight line either. Dr. Scott was connected with some very senior people like Donald Gregg, Eugene Braunwald, who was director of NHLBI at the time and was just moving to open up the Department of Medicine at the new University of California, San Diego Medical School. They were just starting to open that. So I went down and interviewed for a postdoc fellowship with both Gregg and Braunwald. They both offered me a position. I went so far as to write an NIH grant, which was funded. And then a young fellow in the department named Bob Alteveer, who came from Indiana. He was a Dutchman, had come to the U.S., worked with the D.B. Dill group that was in its waning years at the time. And he joined the department, and he said to me, Alan, you know, that's just going to be doing more of the same thing. And he knew I talked a lot about wanting to apply cybernetics, and I was interested in computers. That whole area was just beginning to emerge. And he said, you know, you really ought to think about Arthur Guyton. And I said, well, okay. He said, he's the most creative thinker in the field right now. And he said, if you haven't read his monograph, just read it before you make any decision. And I said, well, I have kind of looked at his monograph, actually. It's been on the shelf, and I thought it was too mathematical for me. And I'm a little afraid of it. Long story short, as I thought about it, I figured, you know, maybe I can learn math and
You did economics.
do some things. Yeah. And it turns out retrospectively, I think the thing I enjoyed about economics was the whole business of economic control theory and the application of computers and cybernetics to economics was just evolving when I was in college, and that's the part I enjoyed, along with the history of economics, which I enjoy. I love history. And so I think retrospectively, in the back of my mind, I've always been interested in systems, the regulation of complex systems. And that attracted me very much to what Guyton was doing. So I got on a plane, went down to Jackson alone. He greeted me at the airport, put me in his old station wagon, which he had rigged. For those that don't know, he had gotten polio during a surgical residency at Harvard, at Mass General during the war, and couldn't operate anymore, and went to Jackson, where his father was the dean of the medical school at the time. It was a 2-year medical school up in Oxford, Mississippi. And by the time I arrived, he had been there for more than 10, 12, 15 years probably, since the mid-50s. And he was the warmest person you can ever imagine. He was soft-spoken. He put me in the station wagon, and his wife Ruth was with him. And he took me for a drive, all up around the northern part of the city and out to the Lake Barnett Reservoir, which is about a 20-mile long reservoir perched above the city. And talking to me the whole time. In retrospect, he must have been picking my brain to see whether I would fit there. And stopped in the evening and took me to a fish fry. There
Had some catfish?
I had some catfish up at the reservoir. And then was just wonderful to me the next day. He just sat and talked, and this is the way it went for the 12 years I interacted with him. He'd sit and just throw out ideas, and think. He had unique ability to define a problem. And then he had a drawer full of ideas on how to, how to experimentally solve that problem. And he put me in a lab directly attached to his office when I first went down as a postdoctoral fellow. I went as a postdoctoral fellow thinking I'd be there for about 2 years.
Before you go on, how was your wife Terry about moving to Jackson, Mississippi?
Well, given the fact that Braunwald had offered me a place in sunny San Diego, she thought, I think, I was a bit odd, if she hadn't already figured that out.
Right.
This is one of the most important people of my life. I met her at Hahnemann. She was working as a technician in the Department of Anatomy for a guy named Ray Truex, who was chair of the department. She was doing histological sections of the central nervous system and lower brainstem on humans. Had worked with whale hearts, the Purkinje system, and she was a real lab rat. And she understood all that. And they put me in her laboratory. You have to ask me what the question was again. I'm diverting from Guyton to my wife.
Well, I brought it up.
Both two very important figures in my life. And I didn't have much room for the graduate students to do the neuroanatomy and histology. She had the microscopes in her lab. So the chairman said, well, you can use one of the microscopes in Terry's lab. So I did. She was not happy having a student in the lab. And this was not a smooth relationship for about half the year. I was resented there because I knew when she was coming and going, and she didn't come early to work, you see. And I didn't have a key, so I'd have to go to the chairman to open her lab. Matter of sensitivity. Long story short. By the time I had finished my master's degree, we'd gotten to be good friends. And we got married. And it was clear, it was a great deviation for me to have a girlfriend that actually understood what I was doing and what science and lab work was all about. Besides having all the other wonderful attributes she has, and being a great deal of fun, and a really deep thinker about a lot of things. And it was just one of those, another lucky turn in my life. So she went down with, it was not a big argument, it was just a sort of questioning why I would do that. And I did my best to explain to her, and she understood and appreciated that. But I wasn't entirely honest because I thought it would be 2 years.
Right.
Well, I was honest. I honestly thought it would be about 2 years, and then I'd find a job somewhere. It was the middle of the Vietnam War. There There were not a lot of jobs. I was really enjoying my work with Guyton. We really clicked, worked well together. It was a clash of the cultures in many ways.
Mm-hmm.
Anybody that knows my wife, Terry, knows she's very liberal. She got very much involved in the civil rights activities at the time in Mississippi. Martin Luther King had been shot just 2 months before I graduated and before we went down there. And she really got engaged in that. And I thought, well, you know, that's it. Dr. Guyton will find out she's a rabble-rouser and say thank you very much. He never did. And he and Terry actually became just the closest of friends. They would often have fellows and students out to the house. He'd always reserve a place for Terry next to him. And they would have dueling arguments about mostly social issues.
Okay.
All through the evening. And he just loved it because nobody would challenge him. People were afraid of Arthur Guyton. You never won an argument easily with Guyton. He was one of the most logical thinkers I've ever encountered. He was an incredible debater.
Mm-hmm.
Very quietly, never raised his voice, just this back and forth, very quiet discussion sort of debate. But he could corner you really quickly.
Well, you were down there for, what, about 12 years? I
was actually there. First, I went down in 1968, and in 1974, I got an Established Investigatorship Award from the American Heart. At that time, you could take these anywhere, in Europe or U.S., into a sabbatical year. I decided, Terry had started college at Millsaps by that time.
Mm-hmm.
And she was a straight A student, and I said, well, you know, you're interested in political science. If you apply to the Radcliffe-Harvard program and get in, which I was thinking that's not a very likely scenario, I will spend the year in Boston on the sabbatical year. She applied, she got in, and won very few transfer students into the program. So I went and spent the year with Cliff Barger. Arthur called Cliff. They were very close, of course, both very active in the American Physiological Society.
Both former presidents.
Knew each other very well. So he just picked up the phone and he said, Cliff, Alan would like to spend the year with you. So I did, and that was actually a wonderful experience too. I met a lot of people there, interacted with people of my vintage that became lifelong colleagues. Victor Zhao was there with me at the time. Mark Fishman was in the lab at the time. Also Ed Haber. I was developing my vasopressin radioimmunoassay at the time, and Ed Haber was one of the paradigms of, he had developed the Renal assay and antibody development, monoclonals. He was leading the field. He was over at Mass General, so I'd go back and forth between the C building and the Harvard, at the Harvard Medical School, back and forth between Cliff's lab and where I actually worked with Claude Lechen, who was doing micropuncture work. Got to be great friends with Claude, and also with Ed Haber. And Ed Haber, that was a wonderful relationship because Ed was very curious about Guyton's theories of cardiac output and venous return and arterial pressure regulation. So when I went to meet Ed, it was a pretty imposing cardiology professor, actually. He scared a lot of people.
But you had to deal with Arthur Guyton too, so you were used to being scared.
I was not easily intimidated. And I was really grateful when Ed said, come into my office. He said, I'll make a deal with you. He said, I'll teach you everything I know about liquid chromatography and antibodies. If you teach me everything you know about regulation of cardiovascular function, and Guyton's theory. I really want to understand this theory of arterial blood pressure regulation. And so I'd go over there 1 or 2 days a week, and on one of those afternoons, Ed set aside a couple hours, and we would just go in his office and talk science, which was wonderful. And those experiences, even though it was a single year, I interacted with just a lot of good people. Cliff and I, and the family. I mean,
Right.
Cliff really took you in as his family, and Claire remains our wonderful friend. It was a wonderful year, actually.
You went back to Jackson. By then you were halfway through your period there. Tell us a little bit about the people you worked with, both in your lab and the people who were in the department. And the other question I have, and it's always been an interesting question to think about, is Guyton and his protégés were oftentimes presidents of the American Physiological Society, and we can get to that a little bit later, but I think it's one of the interesting aspects of his legacy.
Yeah, it is. So who was there and why did all that happen? I think one reason it happened was because Guyton was so active in the society. And this was the go-to meeting, the APS summer meetings at the time, fall meetings, usually on campuses, and the Atlantic City
FASA
meetings. And he'd load up, he would drive, and everybody would pack up and get in the car, and the whole department would drive to Atlantic City from Jackson. And so a lot revolved around all of that. And in the process, it just got ingrained in everybody that this was our society, and we were responsible for its future and making it work. Vernon Bishop was there when I arrived. Aubrey Taylor was there. Harris Granger was there. Gabby Navarre was in the lab right next to me. All of whom,
except for Harris.
all of whom but Harris.
He was the
became
editor
president.
of AJP.
Right. But all involved deeply in APS through their lifetime. Who am I missing? Alvin Smith was there. He was not so active in physiology after he took a deanship over in Texas somewhere. Tom Coleman was there. He was one of my contemporaries, a computer modeler that I learned an enormous amount from. When I went down there, I dedicated myself to computer modeling for the first 2 years. Tom Milhorn was there, engineer, had written a book already, a young faculty member. Tom Coleman was there, and Arthur Guyton, and they were really leading the efforts in all the computer modeling. He had an old analog computer. It was the size of half the wall here, left over from the Korean War that he had picked up in surplus. And it was the most intimidating thing you would ever imagine. It had just hundreds and hundreds of large potentiometers in the front, and it looked like a telephone, an old-fashioned telephone switchboard with wires going in, connecting this and that. And then you'd pull out these large trays of potentiometers with the big ones on the front, and there were dozens and dozens of smaller ones, and then smaller ones, and there were hundreds of these things. And so I first, he gave me a project. He said, well, go develop the electrical analog for the baroreceptor reflex system, which I did, long story short, and I programmed it on this old analog computer. About the end of my first year, he said, well, okay, so that was a good experience for you, but analog computers are out now. We can actually buy a digital computer, and I've been saving up money to do that. And it's an old DEC, and it wasn't old at the time, it's a DEC 9 system. Brought it in, and that was about the second or third year I was there. And then I started to learn how to program it with Fortran, did some modeling. And then I realized about that time that I really was not cut out to be competitive in that area. I was working alongside Tom Coleman, and I thought, boy, I'll never get to his level. Mm-hmm. What I really do best is at the bench. Conceptual thinking, I did well. Bench research, I loved. I loved to work with my hands, still do, and that's what I enjoyed most. So I really began to focus on the bench laboratory research and work with the bioengineers, which was a great lesson in my life, which I've carried forward, and that is just surround yourself. You can't be good at everything. And I learned to surround myself with people I could collaborate with and pursue my interest in those areas and use those to my advantage to understand physiological systems. And that I've done since I've moved to Milwaukee. I mean, that's how I built this department. I brought in people like that. Andy Green, trained in biomedical engineering at Hopkins. I brought Billy Barber up with me from Jackson, who had been trained as an engineer. So I always appreciated the importance of the quantitative aspects and the needs for quantitation in physiology, which has traditionally been weak and has gotten weaker in many ways. And that's one of the great lessons I learned working with Arthur Guyton and all that computer modeling, even though I've never been a great modeler myself. I've always used it as the underpinning for the way I think, and I've always tried to interact with these quantitative people.
In 1980, you came here to the Medical College of Wisconsin. You were a full professor in Jackson. What made you decide it was time to become a chairman? And tell us a little bit about what the department was like here vis-a-vis what you left in Jackson, Mississippi.
So what made me decide to do that was my promise to Terry. My 2 years had grown into 12. And culturally, we were not the best fit in Jackson. And she and I knew that would always be the case. I could have stayed. I think Dr. Guyton was disappointed that I left. I know he was, but he understood also. And so I started looking for positions, chair positions. I was too young to be a professor. That was his trick. He promoted me quickly, so it was hard for me to do a lateral move. And my only move up would be from professor to chairman.
Interesting.
And I had been, at that time, he promoted me before I went to Boston to professor.
Okay.
Thinking I may never return, although I was legally obligated to the American Heart to return. And I wanted to return. So I started looking at chairs in about 1978, 79, interviewed in a number of places in the South, Emory, Gainesville, and then Terry decided, no, we wanted to get a little further north. So I interviewed at Mount Sinai, New York. And no, I think those are the three I interviewed chair positions for. And then I was at a meeting in Mexico City, an Inter-American Society of Hypertension meeting. And then I learned that there was a position in Milwaukee at a place called the Medical College of Wisconsin. And I'd never heard of the place. He said, well, it used to be the Marquette Medical School. And I said, well, I've heard of Marquette University. So I was in a meeting in New York, and on the way back I figured, well, I'll circle by Milwaukee and see what it's like, and I did. And they had just moved out from downtown, the Marquette downtown campus, into a new building up here in 1978, and this was then 79. and there was not much going on in the building, except the faculty was really nice. It was a really open collegial group of faculty, not doing a lot of research. Jim Smith was the chairman. He had retired 2 years before, and the search had gone on for 2 years, failed search, and then this was the second search that they started. And long story short, I brought Terry up, and she liked it. It had more of the flavor of what she was used to in the Northeast. It was ethnically very diverse, and it was a place she thought she could live. And so I said, okay, let's give it a try. I said, well, I'll give it a try for 5 years. I was 39, going on 40 at the time, so I figured, well, you know, if it doesn't work out, I'm still young enough to move somewhere. And so I moved into the 5th floor, which you've spent some time at today.
Yes.
At the time, it was the new building. It was the research building on the new campus out here. And there were 2 people with grants on the floor. One was Hank Adelhauser, who actually had a program project grant on corneal transport mechanisms. Spent the summers up in Cold Spring Harbor, took his lab up there, a wonderful guy. Later went to Emory and set up the program and research in eye down there for them, head of the Eye Institute down there. And the other was Bill Stakeel and Julian Lombard, who had just finished the postdoctoral fellowship. He had gotten his degree in the department with Bill Stakeel, who was interested in the sympathetic regulation of the circulation. And Julian had been a graduate student in the department and went up to the University of Virginia to work with Brian Dooling. And he had just returned, and he and Bill had a grant. And that was about the extent of the research, except for J.J. Smith, who had always done human research and was working, his research was over at the VA hospital. And so I thought, having had a conversation with a wonderful dean who really sold me on the place, Ed Lennon, who later became president of MCW. He was a nephrologist. He and Jack Lemon, it was Ed Lennon and Jack Lemon, had both been recruited back from Boston. Wonderful guys, both nephrologists doing acid-base balance. And anyway, Ed couldn't provide me a lot, but he said he could provide me 4 slots for faculty recruitment, and he'd buy me a computer.
Ooh.
And he'd do the renovations I needed for my chronic instrumented dogs, which I was studying at the time, and set the space aside for that. And he did that. And although the school didn't have much, I had enormous moral support from the dean. And when I'd bring people through, he'd bent over backwards trying to attend to their needs. So that was an attraction to me. The other places I interviewed, some actually were interested in second visits. I never went back for second visits because they were hidebound classic physiology departments. Had a lot of senior faculty,
Right.
not quite senior enough to retire, not much ability to change and shape anything. And I saw a great opportunity to be able to shape a department the way I thought it should be shaped, which was very unconventional thinking at the time, and that was a highly collaborative, integrated, programmatic directed research program.
You were given 4 slots. Who were the 4 you hired, if you remember?
Well, one was Richard Roman.
Okay.
It was one of my first recruit. Well, I brought one, Billy Barber, so that's one. And then I recruited Rich Roman, who was at Harvard at the time. He had trained with Jack McGiff in Memphis, got his PhD there, then was working with Claude Lechen in the Department of Physiology. After I had got there, actually, but I knew him through Lechen, and
Right.
we actually did some collaborative work together even before I recruited him. So Rich Roman was the second one. And then I recruited Jeff Osborne, who trained with Jerry DeBona at Iowa in renal sympathetic nerve regulation of kidney. And who was the next one? Of course, it was John Francois Liard.
Okay.
John and I had been postdoctoral fellows together in Jackson. John and his wife Fanny had come from Switzerland. He had trained in Bern, did rat work. Guyton put him in my lab to train him how to do dog work.
Okay.
They were there for 2 or 3 years. We became good friends of the family. Our families became good friends. John ultimately, after a stint through the Cleveland Clinic with Ferrario's group also, went back to Switzerland, became director of a cardiovascular institute. And I called John and I said, John, I knew he was not happy with, he was in Freiburg, long story, but he was not happy with the way his life in the institute. So I convinced John to come over. He was the only full professor I recruited. John brought a boatload of instrumentation. He was a wonderful experimentalist, wonderful teacher, got all sorts of teaching awards while he was here. So he was the fourth. Those are the first 4.
Now, clearly you have built one of the stronger departments of physiology in the country. And it sounds like part of the success has been a result of the strong administrative support you had here. But what else do you think has resulted or contributed to the fact that you've been able to build a strong department? What characteristics were you looking for in people?
Collegiality, diversity of talents. I wanted from the get-go to do collaborative research.
Right.
I didn't want a bunch of prima donnas and isolated labs in the department. I knew from the get-go we were not going to be a Harvard. We couldn't recruit prima donnas, and we could only succeed if we worked together. And I needed people that bought into that idea. And that was the underlying principle upon which I recruited people. I explained what my vision was when they came. I said, you know, you're either going to work, doesn't mean you can't have your own ideas. Like you're here because you do have your own ideas, and we need to feed off each other's independent ideas and thinking. So, but we're also going to have a program project grant where we actually work together and share resources, because we don't have a lot of resources. The only way we can bring resources and really compete in this world. And so it was what I insisted on. And if I found even somebody that was a very good scientist, if it looked like they weren't going to fit culturally, I didn't bring them in.
Okay. One of the things that is really amazing about your career, Alan, is the fact that you have been continuously funded since 1971. And as you step down from the position of chair of the department here, you still have research funding, and you're intending to continue to do your research, and I commend you for that. But tell us a little bit about the characteristics you think that have contributed to your ability to sustain funding for so long.
I think I've already mentioned some of them. Surrounding myself with really good, strong colleagues and scientists who could fill in my weaknesses. And practically all of my research has been collaborative. My R01s have been, except for my first one, was collaborative. And that's that's clearly one reason. And the other reason is I've worked very hard. It's hard work.
Mm-hmm.
I never viewed it as hard work, but I guess in retrospect, it was hard work because it's relentless. Mm-hmm. It's very competitive. It's not without its tensions. Trying
mm-hmm.
to get grants renewed is tension in one's life. On the other hand, I've always been inspired by grant and grant writing. To me, a grant deadline is the period, 6 months before the grant deadline is my most inspired period of thinking. I really
mm-hmm.
focus, concentrate my efforts. And that goes on, I just live it 7 days a week.
Right.
And so I don't think there's anything unusual about that. Anybody in science has to work hard, or they're not going to compete in the business. It got harder.
Of late.
It was easier in the 70s. But I must say we've gone through cycles. There have
mm-hmm.
been better cycles and worse cycles. We've been in the worst for the last 10 years, since 2008.
Right.
And it took more work. It took revisions of grants. There were times, but when I got to be 65, I started to ask myself, do I really want to do this? And my answer was always, yeah, because I really enjoyed it. And I really enjoyed the people I was working with. And that's what made it successful. And I think they felt the same.
Yeah.
It was a group of people that really cared about each other, worked with each other, helped each other, and cross-trained our students. We've always had, we didn't have a formal name until it became fashionable. We multi-mentored our students
right.
in many ways because of all the collaborations that were going on. The labs were all open. It's a completely open lab environment. Students wander in and out of everybody's lab, picking up techniques for this, that, and the other thing. And that's just the way the department's grown. And it's what makes it such a joy to work in.
You, in addition to the leadership roles and programs that you've run in Mississippi at MCW, you also took leadership roles in societies. You were president of the American Physiological Society in 1997. You were president of the IUPS in 2001 through 2005. Can you tell us about the important roles society service plays in your career and plays in anybody's career?
Yeah. Obviously, I put a lot of energy into those jobs, as it were, because I felt then, and I continue to feel, that as a young scientist, and certainly as a senior scientist, but particularly when you're starting, you're just lost.
Mm-hmm.
You're lost in the woods. You need a place where you're welcome, where you can get to meet more senior people
mm-hmm.
in the field, established people, other people of your age, struggling with the same problems. It's the only way you really get to understand the universality of science, and the society plays that important role. And I saw that early, and I must give credit to the American Heart Association too, where I was very active in the Council for High Blood Pressure Research. These are the ways that you not only advance your science, but you advance, to me, one of the most rewarding parts of science, and that is you get to know people beyond your immediate sphere. You get to know people from all over the world,
mm-hmm.
National meetings, international meetings, people that you develop with. you compete with, you joust with in terms of concepts, and if you're not part of the society, these are just names on papers. And it's the society that brings all of this together and enables it to happen. It can happen. And the other part of the society, the wonderful part about the APS is that it's a growing, it's not been cast in cement as some societies get cast. It's always churning. And once you're in the society, as it allowed me to do, I went from being a staunch part of the cardiovascular section. And then I realized that a lot of the stuff I was doing was involved in fluid and electrolyte balance as I moved more toward the kidney. So I got in the fluid and electrolyte balance section. And then, of course, we started the Physiological Genomics Working Group, and now I go to all the sections. But each one of those, including the renal section, which I am very much involved with now, have added and expanded my group of colleagues, scientific colleagues and personal colleagues. So it allows you to go in a very fluid way. Yeah, I know there are cliques,
Right.
but the cliques are not that difficult to overcome.
Right.
If they know you're serious, and it takes a while, they have to recognize you're serious about doing something in this field, which was not the one you grew up in. But I've worked in 3, 4 different fields now, and it's been just fine. And that's been one of the most rewarding parts of the society. So it's had an enormous impact on my life.
And your service to APS has been greatly appreciated, and obviously you and I have had a wonderful working relationship over the years.
So let me make a comment about that.
Oh, I'm sorry.
I mean, really, you've been there through most of my professional career as the executive director. And all of this really has to be held together by some very competent person who understands what science is all about.
Well, they got me instead.
And understands, yeah, well, it worked pretty good. So you have been really not only an important part of the American Physiological Society, but an important part of my life.
Agreed.
We'll put it on the record.
All right.
We are real buddies.
I will agree to that. The department here at MCW recently found itself in a book called 1 in a Billion. And it was a story of Nick Volker, and it revolved around much of your research and the research of Howard Jacob. And tell us a little bit about how that all came about. I think it's a very interesting story.
It is an interesting story. I got to know Howard Jacob. I'll start back at his days at Iowa. He was a graduate student of Michael Brody, who was chair of pharmacology. And he was doing neuropharmacology and very prominent in the field of hypertension and the baroreceptor reflex system, which I was very much involved in at the time. And Howard's project was in rats studying the baroreceptor reflexes. His mentor, Mike Brody, took a trip. He was internationally a very prominent guy. He'd been invited to Australia and then took a week vacation up in the Barrier Reef, and unfortunately had a heart attack and died. And Mike and I had been good friends, and we went back and forth between Milwaukee and Iowa, and we had been each other's guests. Well, I'd gotten to know Howard through Mike, and suddenly Howard needed a mentor and an external advisor in his committee. So I essentially mentored Howard till the end of his dissertation. And then I had a graduate student, finishing the same time Howard did, Jose Krieger, who was Eduardo Krieger's son from Brazil, from Sao Paulo, from Ibrahim Preto, actually, and who was also very active in the IUPS and APS, and very prominent in the bioreceptor field. Well, he sent his son up to do his PhD with me, and he was finishing the same time Howard was. And Howard had decided he wanted to learn how to do molecular biology. And I was telling Jose the same thing when he finished. I said, you know, you learned a lot of physiology from me, but you didn't learn much molecular biology, and you're going nowhere without it. And so, long story short, they both ended up as postdoctoral fellows in Victor Zhao's lab in Boston. Again, one of the residues of my connections with Boston. I knew Victor well enough just to pick up the phone and say, I've got a great student here. And Howard. So they both went in there together, both remarkable, outgoing, big personality kind of guys. Perfect for the Zhao lab, actually. So Zhao sent Howard over. He was interested in genomics, getting interested in genomics, and there were no reagents for the rat. There were no markers to begin mapping phenotypes for the rat. So he sent Howard over to work with Eric Lander, and at MIT. Eric and Victor had started a collaboration. It wasn't going nowhere because they didn't have the right postdoctoral fellow to go in and do all this laborious work. Howard went over there, spent nearly 5 or 6 years with Eric, started to raise his family. In the meantime, Jose went back to Brazil, actually. I'm shortening the story, but he ended up back in Brazil. So I was interested at that. I got very interested when the two were working together in where genomics was going. They were just starting the Human Genome Project, and I was trying to figure out how physiology was going to fit in with this. And Howard and Jose and I met at a meeting after had been up there about 2 or 3 years by then, and Howard was getting markers and starting to really make some progress. And we brainstormed at a meeting. It was, again, an international society, it was an Inter-American Society of Hypertension meeting in San Diego. And we left for a coffee break on one of the days, and we're sitting out on the veranda overlooking the beautiful Pacific, and I started picking their brains about what they were going to do with all this. And at this time, Howard said, you know, what I want to do, I can't do at Boston. He said, they don't have physiology anymore. He said, the only way this stuff is useful is to attach function to these genes, and I can't do it there. And I said, well, you could certainly do it in Milwaukee, Howard. And this led to an extended conversation. Long story short, I was able to recruit Howard to MCW, where he was from northern Iowa, as his wife was. They were starting to raise their family. They wanted to be close to the families. So the timing was perfect. We started, even before he left, the program project grant, a specialized center of hypertension research. Howard had gotten to know, through Eric Lander, Pavel Hammett at McGill in Montreal. And he was beginning to work with this isolated population up in Chicoutimi, Lake St. John, about 200 miles north of Montreal. And Howard said, well, I know Pavel. He said, let's talk to Pavel. We could get that population. I said, well, I've got Ted Kotchin here. He's chairman of medicine. He's studying a large African-American population. He and his wife, Jane, said, we'll have a French-Canadian population, an isolate population, an African-American population. Anyway, long story short, we put a program project grant together. I was doing rats, and Howard was going to do rats. And Eric helped us. Lander gave us a lot of advice in putting the grant together in terms of analysis of SIB pairs and things in the human population. We got the grant funded, and that was the beginning of a 20-year career with Howard at MCW. And after about 15 years, Howard began to realize that this had to go to human now. And even though we had developed large programs to develop all sorts of congenic and consomic strains of rats and chromosomal substitution approaches to understand the relationship of genes and function, which is very well funded and very well supported. It was clear as things went on that this was going to move to the human, and we were now getting the tools to do that. And Howard, by that time, was buying and setting up sequencers, no longer running the old gels that we started with. And the people that were interested in sequencing were the people at the Children's Hospital here, rare diseases. Mm-hmm. So Howard got hooked up with the Department of Pediatrics, and very soon really got immersed into this. He decided that this was going to be his life. And we didn't have the resources to do it. At that time, nobody had actually used whole genome sequencing, exome sequencing in this case, to diagnose a human disorder. Everybody was talking about it. A lot of people were trying to do it. This little boy, Volker, was just the right person at the right time. His family was, along with the Children's Hospital, were able to raise whatever it was, about $80,000 or so, to do the sequencing. Howard brought together, and this was all Howard at this point. Mm-hmm. I had nothing to do this except stand in the background and say, "You go."
You got mentioned in the book.
and they were able, actually, he worked with a group of budding bioinformaticists. They weren't even called that at the time, to do all the analysis. They wrote all the original software to look at the sequence variants through the whole genome and got it down to what appeared to be a likely candidate. It turned out it was the likely candidate. It was druggable, and the little boy who had undergone I don't know how much surgery, multiple surgery, his whole GI tract was just dissolving away, and he didn't have long to live. And as soon as they realized what was underlying this, and the gene defect and the protein that was missing, they treated it, and Volker's alive today. This was 10 years ago. And one of the local newspaper, the book drew the attention of our local science writers in the Milwaukee Journal, and they wrote this up in a series of articles, got a Pulitzer Prize for it, and then later wrote a book about it.
There's a very nice story about how basic research leads to clinical application.
Yeah, it's a wonderful story.
Yeah. Let me ask you one or two more questions, because I know we've been going on for a while, but you mentioned Terry. In addition to your being an outstanding scientist and leader, you have been a dedicated and supportive husband of Terry. Mm-hmm. You've been married how many years now?
52.
52 years.
Mm-hmm.
And you have been very active in an organization that she has started, the TMJ Association. And so tell us a little bit about your role and a little bit about Terry's efforts.
Yeah, this is really the strength of Terry. She had some surgery nearly 25 years ago because she had her jaw was flopping, dislocated. And the dentist didn't want to work on her teeth. They said, "We'll only work on your teeth if you get your jaw stabilized." And so ultimately, after really needing a dentist, they convinced her to go in and have a procedure done. At the time, they were going in and replacing these meniscus with artificial silastic and Dacron mesh silastic implants and Teflon implants. They had been trying a lot of things. None of them, it turns out, had been
Safety tested?
yeah, not even in animals.
Yeah.
And I was naive and stupid. It was being done in an academic center. I figured, you know, it must be okay. These oral surgeons must know what they're doing. And it seemed like a relatively innocuous thing, just to go in and replace the disc. It turned out to be not innocuous. It turned out to cause her a lifetime of pain, which she still has, and of anger with the system. And after about a year of being virtually debilitated after the surgery, and being told by the oral surgeon that, you know, you're a very unusual case, this has never happened to us before, she ran a little advertisement in the Milwaukee newspaper saying, if there's anybody out there that has similar problems, some TMJ problems or post-surgical implant problems, I'm going to have a little meeting over at the medical college on whatever, Thursday night, expecting a few people to show up. And she had a room full of people. And at that point she realized that, and the surgeons realized that the cat was out of the bag. She then started bringing home articles from the dental journals about implants and TMJ. And she'd have me read them at night. And I quickly realized there was no science to any of this. In fact, it was just awful. And she decided, well, she was going to try to do something about it. We needed science. And I said, you know, this is impossible. You're fighting the whole dental profession. You're fighting the oral surgeons. You're fighting the NIH that's not supporting research in this area. There is no way that one woman is going to change anything. So I was going to Bethesda for a study section, and she said, can I come along with you? She said, I'd just like to see what the NIH campus looks like, and I want to go in and talk to a few people at the Dental Institute. And so I dropped her off at the front door of the Dental Institute, went to my meeting, picked her up at the end of the day, and oh, it was worse, she said. She says, it's even worse than I imagined. She says, these are the people that are supposed to be thinking about science and thinking about the welfare of patients. They're not. And that led to actually her TMJ association. She got support from an investor in town that originally helped support, set it up, and rented an office for her, and helped support Deanne Clare, who has been with her for 20 years now. And then I volunteered my effort to put scientific meetings together. So I've been writing a NIH grant every other year for the last 15 years. We've had a wonderful meeting, which Marty Frank knows very much about, because given very low resources of the organization, we couldn't afford a big hotel. And Marty made the FASB Conference Center available for every one of these meetings and introduced the meetings and got to know the issues. And it has had an impact. I mean, I think Terry there were a whole day of congressional hearings that Terry initially got pulled together. There were some mandates given to the NIH to do something. When the genome sequencing stuff started, there was a $30 million grant to Bill Maxner's group down at North Carolina. He's at Duke now. So they've got attention to this, but it's been long and slow, and she is just she just won't let go of this, fortunately, and and I'm very, very proud of her. I will do anything to help her in this effort.
Well, I think that's really great, and I think it really does say, in spite of the fact, as you started out, she didn't have a chance in hell of doing anything.
Yeah.
One person with passion can make a difference.
Yeah.
Whether it's in this situation or any situation, dealing with Congress and NIH and FDA and things like that to move things
It's been
forward.
one of the great educations of my life. She's hauled me all over the halls of Congress, House and Senate, talking to people I didn't want to talk with.
Right.
She's learned how to work both sides of the aisle, and she puts all of her politics aside for the needs of the patient, and it's been a great education to me.
Alan, you've had a great career. From my perspective, I know you think you've had a career that you had not expected when you started, and it's been highly successful. But the last question I ask is really an important question for any early career scientist starting out, is really, what is your advice to students starting out in a career in science or physiology, if we would prefer?
That's not an easy question to answer. I think there are a few general things, though. I think I would advise them to follow their passion, what excites them in the science. If they don't get excited about anything, they're in the wrong field.
Mm-hmm.
But something must have attracted them into it. That's one critical thing. And that's their ability to follow through on what interests them in science is very dependent on the mentoring, who they end up with, because there are not all that many options. And so pick your mentor very carefully. Be prepared to work very hard. Be prepared to have ups and downs.
Mm-hmm.
There's a lot of downs, a lot of blind alleys in science. But there's also incredible excitement and joy when you actually discover something that has not been seen before. And I think above all else, remain true to yourself. Honesty and collegiality are critical elements of science. Nobody goes it alone. I don't think they ever did. Mm-hmm. Even though they may award separate Nobel Prizes. If you dig into the careers of these people, they're just one of us.
Right.
And it took a lot of interaction, a lot of patience. It takes some luck, and you have to be prepared for high periods and low periods. But if you don't really have a passion and enjoy coming to work every day, that's a problem. You have to really enjoy coming to work.
Thank you. Alan, let me thank you first for being willing to tell your story as part of the Society's Living History program. Second, let me thank you for your many years of friendship as well as leadership in the Society. I wish you great success after you step down as chair and devote your whole energy to your research program. And I look forward to many more successes from you. Thank you, Alan.
Marty, thank you very much. I'm honored.
Thanks.